Hi, Mr. Wartsky here again, and we are going to talk about the cell membrane today, or start to talk about the cell membrane. Remember, if you have any questions, you can either ask me in class, or you can review the video at your own leisure, look in your textbook, and you can also pause when you need to, so you can get some notes. So today we're going to focus our attention on the cell membrane. I'm going to tell you that it is a fluid mosaic model. Okay, what does that mean? Well, let's first talk about what makes up the cell membrane. First of all, it surrounds the cell, and the main components are phospholipids, and the phospholipids are these things that have the round head and the two tails. Hopefully you remember that from the organic molecule material that we covered. The proteins are these blue things here. We have different types of proteins. Cholesterol are these they have ringed molecules there, and they actually serve a function in the cell membrane, very important. Glycolipids as well as glycoproteins. And you can find the glyco is here are the carbohydrates. The carbohydrates can be attached to the proteins, or they can be attached to lipids right here. So if you need to, it might be good to get a simplified drawing of that down in your notes. Hit pause. So the fluid part of the fluid mosaic model, basically it is not static. The cell membrane is constantly changing, moving around, and the fluid is maintained, maintains its fluidity. How? Well, cholesterol is the molecule that helps the cell membrane maintain its fluidity. In warmer temperatures, the cholesterol slows down the movement of the phospholipids. In colder temperatures, the phospholipids tend to pack together and move slowly, but the cholesterol prevents them from packing too close together. So the cholesterol helps the cell membrane maintain its fluidity. So you need cholesterol in your diet. So here is a little bit of the phospholipid bilayer. We have one layer up here, one layer down here. The phosphate is on top. These are the two lipids. In the middle here, this is hydrophobic, while the outside is hydrophilic. So the mosaic part, there are proteins in the membrane. Okay, so there are different types of proteins. First, we have the integral proteins, which are actually in the membrane. And there's two types of integral proteins. We have unilateral, which go part of the way across, and transmembrane, which go all the way through the phospholipid bilayer. The second type of proteins, the major type of protein, is peripheral. They're held on the outside or the inside of the cell membrane. If it's on the inside, then the cytoskeleton, which we just talked about, holds it in place. Now, carbohydrates can act as receptors as well as the proteins, which allow cells to recognize other cells. So here's another look at a phospholipid. Kind of crazy, but we have all sorts of stuff. We have the cholesterol. You take a look at that. We have the phospholipids, the glycoproteins, which are here, glycolipids, which are here. There's a glycoprotein. Protein goes all the way through. That'd be a transmembrane part of the way through. We don't have them around here. Bummer. They all look like transmembranes to me. And we have unilateral. There's no peripherals. Okay, so the cell membrane is a selectively permeable barrier, which means that it selects what enters and exits the cell. There are 7 ways that substances can enter and exit the cell, and we're going to talk about each and every way. The first is bulk flow. In bulk flow, pressure, water pressure, hydrostatic pressure, forces the molecules through the cell membrane and into the cell. It basically pushes through. No matter what's going on, the pressure is so great, the molecule is going to enter. Diffusion is the movement of molecules from a high concentration to a low concentration, and this can only be done with small molecules that are hydrophobic because they have to be able to get through the hydrophobic interior of the cell membrane. If it has a charge, if it's polar, or if it's hydrophilic, it will not be able to move through the cell membrane by diffusion. Osmosis, and we're going one of our labs, it focuses on osmosis as well as diffusion. So osmosis, and you need to write this down, is the movement of water from a high water potential to a low water potential through a selectively permeable membrane. As a 9th or 10th grader, they did not give you the whole definition of osmosis, and today we're going to do that. So the movement of water from a high water potential to a low water potential through a selectively permeable membrane. What is water potential? It is the potential for water to move. Pure water, water with nothing in it, has the highest potential. It's the highest potential energy, the potential to move from one place to the other. When you add solute, you lower the water potential. So the more solute, the lower the water potential. When we talk about osmosis, we need to focus on three osmotic environments: isotonic or isoosmotic, hypertonic, hyperosmotic, or hypotonic and hypoosmotic. And that is not a typo. There are actually two words, two O's there. Hypoosmotic. Isotonic. So when the water potential inside and outside of the cell is equal, the water will move back and forth equally into and out of the cell, and the mass of the cell will not change. This only happens when the water potential on the outside of the cell and the inside of the cell are equal. Now remember, we're talking about osmotic environments. These are the environments that surround cells. Hypertonic. In hypertonic, the water potential inside the cell is higher than the water potential outside of the cell. So water flows from a high water potential to a low water potential, so the water flows out of the cell and the cell will lose mass. So what makes the water potential higher outside of the cell? Lower outside of the cell, right? The water potential is lower on the outside of the cell. What makes that water potential lower on the outside of the cell? That means there's a higher concentration of solute outside of the cell. So the water will flow out of the cell in order to help equalize the water potential. So here is a picture. Here we have water molecules, but we have a higher concentration of solute on the outside than in the inside of the cell. That means that the water potential on the outside is lower. The water will flow from an area of high water potential to an area of low water potential, and will flow out of the cell. Hypotonic means that the water potential inside of the cell is lower than the outside of the cell. Since water flows from a high water potential to a low water potential, the water flows into the cell, which increases the mass of the cell. Remember, water always flows from hypo to hypertonic. It's always O to Er. So what do we have for a diagram? Here we go. On the inside of the cell, we have a high number of solute molecules. On the outside, we have a low number of solute molecules. That means that the water potential inside the cell is lower than the water potential outside of the cell, so the water flows into the cell, and the cell will gain mass. Hopefully you got all that. From hypo to hyper. So the osmotic pressure is the pressure required to stop osmosis. In a plant cell has a cellulose cell wall. Now, water molecules keep on going into the plant cell because the plant cell is hypertonic. Well, it's in a hypotonic environment in which the cell has a higher solute concentration or a low water potential compared to the environment. So water constantly enters the plant cell. There's going to come a time, though, when no more water can enter the plant cell because the plant cell is full. Now, an animal cell, when that happens, it will blow up. It will explode. However, plant cells have that cell wall that surrounds it, and that cell wall will push back and prevent the water from entering. When that happens, that is osmotic pressure. It's also called turgor pressure, in which that allows the plant cell to be full, allows the plant cell to be nice and plump and stand erect and be good. If you have a wilty plant, that means there's a loss of turgor pressure because the water is no longer entering the plant cells and pushing against the plant cell wall. So the fourth way a substance will enter is called facilitated diffusion. Now, facilitated diffusion or facilitated transport is diffusion of molecules from a high concentration to a low concentration through a transport protein or protein tunnels. These are protein tunnels here. Yeah, we can see them. Now, these polar molecules will go through those protein tunnels and into the cell. Now, these protein tunnels are very selective, also called permeases. Right, so each molecule has its own specific protein tunnel. Active transport. Now, this transport is special in that it requires energy, requires ATP to move the molecules. It also moves the molecules from a low concentration to a high concentration. Now, you got to think about that for a second because it seems right, but that is against the concentration gradient. Most things move from a high concentration to a low concentration, but not in active transport. It's the opposite. So these molecules will fit into, here's a molecule here, it's on the outside. It fits into this transport protein, energy is used, and then the molecule is dropped off inside the, or on the other side of the cell membrane, and it is dropped off to an area of a high concentration of molecules as opposed to a low concentration of molecules. That is active transport. Vesicle-mediated transport only happens in eukaryotes, and I hesitate to even bring this up, but we'll do it anyway. So there's two types of vesicle-mediated transport. There's phagocytosis and penocytosis. Phagocytosis is cell eating, penocytosis is cell drinking. It basically happens in the same way, but phagocytosis takes in food, while penocytosis takes in H2O or water. So in this, the cell is forming vesicles or vacuoles. We have here food taken in, right? Digest is broken down, and then it'll let out. Receptor-mediated transport, there are cell receptors here, and they bind to molecules, which are called ligands. When that happens, the cell membrane starts to pinch in and forms a vesicle there, and we form a vesicle that is receptor-mediated transport. We have receptors which form a vesicle, and that also requires energy. And that is it for the cell membrane. Again, if you need help, if I move too quickly, hit the pause button, or you can watch it over again. It's up to you. I hope that helped. Have a good night.